Hello everyone, thank you very much for coming back from lunch. It is warmer in here, so there is an incentive to be in here rather than outside. We have our longest session now, where we go into monitoring methods. And to run this session, we're very, very pleased to have Alison Specht. So Dr. Specht has been with Tern since the beginning. She took a short trip out, just found out that the world wasn't as good outside of Tern as it is in Tern. And anywhere you go in the world and meet with people, all you have to do is say Alison Specht, and you've immediately got common ground with every researcher in the world, because Alison is most probably the best networked researcher that I've come across. In Tern, she started out running our synthesis centre, and I think that gives you a hint as to why she is so well networked. She brings people from many different disciplines and domains and institutions together and gets them talking so that they can work out how collectively they can have impact. And Alison's been playing a very large part in what's called the Belmont Forum of recent years, and that's put her on a world stage. But I'll let Alison now take over so that we can get on with our session. Thank you.
Well, it is a very long session, so I will give a very brief introduction, and then each of the speakers will come up sequentially without any prompting at all from me, which is great. But we have a great diversity of topics that are going to be talked about today in terms of monitoring and how you can do it better, from groundwater-dependent ecosystems and groundwater monitoring, to bioacoustic monitoring, to IGSN sample collections. If you don't know what they are, look it up. They're geological and specimen samples and how you can register them over time, and right through to drone work and ex-nearshore work. But anyway, so the first speaker today will be Juan Carlos Montes-Herrera from the University of Tasmania. And we have, I won't call him up quite yet, but we have almost every part of the continent and, in fact, overseas mentioned today, involved in this session, because we have a speaker from the National University of Taiwan who will also speak on some really nice topics about, not spectrophotometry, which is really interesting. So, but before any further ado, we'll very much welcome Juan Carlos. Thank you.
Thanks, Alison. Hi, everyone. It's an absolute honor to be here. I am part of the University of Tasmania, working with Professor Arco Lucier and Professor Ben Sparrow on using uncrewed aerial vehicles as an observation tool for Tern surveillance monitoring plots. I joined this project early last year. I kind of started the building on top of some protocols that were already developed, but I had to learn about what's Tern, what's surveillance. And what I want to do today is to give you some of a summary of that operational procedures and how we feel that we are at the moment operational, from data collection, data processing, to data access. But first, I put this image in here. This is a false color composite of one of our first monitoring plots that we were collecting independently without the bosses around. Me and Carla Franson, the UAV lead at Tern Surveillance. I talk more about what false color composites are, but this is a reminder to myself and hopefully to you of what a different perspective aerial imagery can provide on nature and landscape, and how limited our eyes are. And when you start to collect infrared image, for example, you can start to see the other processes that are happening around. So in this case, there's a fire scar, just as an example. This is a fire scar from a recent fire. And then you can see some recent regrowth in there in this bright yellow-green. And when you notice that, then you can start to see like other fire bites in the landscape and then some other unaffected regions. And I think that's quite a nice brief example of how aerial imagery can give you this spatial context. But more into the science. So drones, basically, or uncrewed aerial vehicles, are a link between field observations. I like the term that someone used of boots on the ground work, and satellite and aerial, like aircraft observations. This to expand our sampling range. Of course, our boots on the ground are limited by our human scale capacity. And then we can expand this sampling range to kilometers and multiple kilometers across a field survey with fine-grained data. And when I'm talking about fine-grained data, I'm not talking about that spatial resolution that comes with aerial imagery that is closer to the ground, not like more pixels, but also with fine-grained data that comes from field observations, that then this validates a lot of the remote sensing metrics that we are collecting both in space and time. To bridge these spatial and temporal changes to reach management goals, identify biodiversity hotspots or areas that going rapid change. So I think this is where Tern comes in, so with this multi-scale monitoring framework, and it's quite a nice opportunity. So I like to think about this project of linking the regional scale monitoring with Tern ecosystem surveillance and linking the Tern landscape for a more continental scale approach. There was a recent talk about the Blue Mountains where they are also using UAVs, but more on a local to regional scale, and I think that this project is more on the regional to continental scale. So if anyone has seen the Tern ecosystem surveillance plot, so it's a long-term monitoring framework where there's like 100 by 100 monitoring plots across all the diverse bioregions of Australia. And it's quite a lot of work, and it's the challenge of our project is to make these observations, these drone observations, in a standardized format and manner that can help us to capture the inherent variability in different bioregions without compromising any important metrics. So that's where this project comes in. So we want to develop standardized protocols for a plot-based drone data collection and processing. And at the moment, we are using two flights. So every plot, we have 2 flights with optical imagery sensors that I'll talk more about later, and LiDAR sensors. So this is the optical imagery platform. It's basically an M300, so it's like a 25 kilo license UAV. And it's carrying 2 sensors, red, green, and blue. So this is an RGB sensor, commonly called. It's like the phone camera, like a phone camera is RGB, but this helps us to link that human eye observation and say, like, oh yeah, that's a tree, that's a shrub, that's a grass, etc., with a very fine spatial resolution. And then we have a 10-band multispectral sensor. You don't hear this multispectral word anymore, but most of our satellites where we get like NDVI, time series change, pigment indices, they are multispectral satellites and multispectral sensors. And the cool thing about this sensor is that it matches the wavelengths that those passive satellites are collecting. So then we can start to link these spatial temporal scales of a fine resolution with our UAV observation, multispectral data, with long-term series satellite data sets. Moving to the LiDAR, that someone already showed it, but I presented briefly. So it's again like the latest models of a consumer-grade LiDAR sensor. So we are using penta-return. I'll talk more about why that's important, getting approximately 600 points per meter square at a 50-meter flight. This is all geo-reference using a DRTK station to get centimeter level positioning. And then first we set up the DRTK station, we prepare the UAVs to fly, and then we fly. While we're doing the flight, though, and this is where I think that Tern has this infrastructure already built that we have to take the most advantage of, is the monitor app that the team has been amazing to develop this module only for UAVs, where we collect who is collecting the data anyway, who is collecting the data? The sky code, environmental conditions, wind speed, and other metrics that I'll talk more about later. But the important is the who, because then it can come back to us who's collecting the data, and maybe we made an error and we can trace it back. And this is important to talk about the UAV team that is currently collecting data at the moment in the Pilbara region, Carla and Troy. So these are just some photos of the fieldwork that we've done, where we have like both, we have a very, like by the 5th day of fieldwork, like it's a very smooth process. Like one prepares the aircraft before flying, and then the other one's collecting the data, preparing the other sensor, etc. So this is a funny plot where we had to trolley in. So we noticed that we can put all our equipment in a small trolley. But shout out to Carla and Troy, who are collecting data at the moment. So but this is much of what we do. So it's like the drone is flying, and there's a lot of waiting, making sure that the data is being collected properly. So if we go back to this slide for the last time, we can summarize our data collection per plot. So for the imagery is an RGB plus 10-baud multispectral, which are basically a bunch of JPEGs and a bunch of TIFF files, which is like a raw image format. And for LiDAR, we are collecting at pentareturn, and these are like complicated 3D files and some JPEG images as well. And per day, we do some data management, so we collect between 3 to 5 plots per day, and we arrange them in this data folder structure. If anyone knows how to, like a more exacting way to present like a data folder structure, but thanks to Aaron for helping me with this. So when the data is available at the moment, so this data is available now in the Tern data portal, where we have the plot ID based on the Tern surveillance monitoring framework, the visit date, imagery, RGB and multispectral, and then a level 0 raw folder, and then the LiDAR with the level 0 raw folder. We also back up the DRTK logs and a metadata folder that we'll talk a bit more about later. So that data is available right now, but there's a lot of research and what we want to get into level 1 data analysis ready products, but this requires the seal of approval of ARCO and BEN to say like, okay, it's ready to be called a data analysis ready, you can publish it, but there's still a lot of work to do here. But in general, for the optical imagery, we do structure for motion photogrammetry using Agisoft Metashape, where we basically produce 2 files, an RGB orthomosaic, which is like 1 centimeter resolution per pixel on a 300 by 300 plot, and a 10-band orthomosaic file. So this is the file that is collecting, that has all those wavelengths in the same image. So we are trying to automate this to make this as co-aligned spatially as accurately as possible. And this is just an example of all the other false color composite. I didn't have space to put 10 bands in there, but this is just an example of the combinations that you can get with multispectral sensors that are related to, for example, photosynthesis, water content, pigment content, etc. Then for LiDAR, I'll talk very briefly. I saw Sean Levick there, so I'll be very, very shallow. But we are using DJI Terra to produce a LAS file and a polygon file. So that's part of our level 1 data. To produce outputs such as canopy height, canopy density, and terrain model. These outputs are then converted into a cloud-optimized format. So I talk more about that at the end. So just to make them available to be in the Tern data portal. These then get into Stack, which is basically a catalog for the data. And when I talk about the metadata thing, this is part of that field monitoring app where we're collecting the observers, but also the flying time, who flew it, any flight settings that changed. This is the available data at the moment, so we're still collecting more at the Pilbara, but We have 140 out of 1,000, so we're still a lot to go. These are just examples of the quality of the data, some more false color composites. And I won't show you those videos, but these are examples of the LiDAR point clouds, the detail of the orthomosaics. But I wanted to get to this one, which is the atoll forest sites, where there's where we're trying to push the capacity of this LiDAR sensor to see, can we collect metrics accurately from tall forest sites? So these are tall forest sites, like 40 meters. And where you can get, if you look at the bottom graph where you have the return, the first return, which is that first laser pulse being bounced back from the canopy, and then the last returns going back, going to the trunk and to the soil. So we think there's like a good potential for this sensor to be used. And then just briefly, this is some work in progress that we're trying to build a web spatial platform that captures this rich environmental data. But that I think this is my last slide. I just wanted to show you the capacity of this. Yeah. So thanks so much.
Good afternoon, everyone. Let's switch channel to the Northern Hemisphere. I'm Kuihan Chu. I'm a master's student from the Department of Geography at National Taiwan University. Today, I will be presenting our research title, Seasonal Drought Alters Avian Soundscape in Taiwan's Mountain Tea Forest Mosaic. And this project was collaborated with Dr. Madeline Goddard and Dr. Cho-Ying Huang. And let's talk about the research background. Climate and land use change are really changing agriculture and mountain ecosystem. In Taiwan's Alison area, tea plantation have been expanding, breaking up to the cloudy forest into smaller patches. Then we can see the line graph below. In early 2023, Alison, we saw a very serious drought. February rainfall dropped to just 20 millimeters, far below the usual average of 112 millimeters. These data are from the Central Weather Bureau Alison Station. And the birds are great indicators of this change because their vocal activity reflects what's going on in the environment. But the big question is, how do their sounds respond to extreme drought, especially in a landscape that's so mixed and fragmented, like this tea forest mosaic? From past studies, we know that climate has a strong influence on soundscape, but what's still not clear is how seasonal drought affects bird sounds in different types of environment. So in this study, we focus two questions. First, how does seasonal drought affect avian soundscape diversity and activity? And second, does the structure of the vegetation help buffer or make the drought effect worse? We are especially interested in places like Alison, where tea plantation and forests are mixed and have very different structure. We want to see how drought affects both nature and farming in this complex landscape. And by the way, the tea quality here is in the world class, so it's good. To answer these questions, we combine several types of long-term data. First, including weather data, UAV LiDAR scan of the vegetation structure, and sound recording from the field. We focus on fine soundscape indicators as our main outcomes and use factors like drought, vegetation capacity, temperature, humidity, sunlight, and terrain feature as predictor. So to analyze all this, we apply linear mixed effect model, which allow us to look at both the main and interaction effect and how these variables interact. especially the interaction between drought and the vegetation capacity. Okay. We collect methodological data and acoustic data from 15 sites in Alison region, range from 900 to 1,500 meters above the sea level, over 4 consecutive Februaries, from 2022 to 2025. We use SM4 audio recorder to record sound 2 hours after sunrise every morning. And it's an example of our sound record of site JP. Let's give it a listen. Yeah, it sounds like this. And we also acquired high-resolution UAV LiDAR data to calculate vegetation capacity metric, like VCI or RI. This part is an example of NF site. On the side, we can see the temporal patterns of 2 key soundscape indices, ACI and BI, across different sites and months. And let's talk about the 5 soundscape indicators we use. First, ACI, capture temporal variation in sound linked to the bird song complexity. And second, the BI represent overall biotic sound energy. ADI and AEI, spiritual diversity and evenness across frequency. And the final, NDSI, measure the balance between biological and human-made sounds. Among all these, ACI and BI we use to assess vocal activity, and ADI and AEI provide insights into spatial structure. Before I talk about the climate effect, it's worth mention that terrain features like elevation, slope, and aspect didn't show significant effects in our model. This might be because the elevation difference across our sites weren't big enough to create strong environmental variation. Now we can look in the result. Temperature and humidity consistent had significant positive effect on most of the acute indices. In particular, BI and ACI, which reflect vocal activity, increase in warmer and more humid conditions. On the other hand, drought only had a clear negative effect on BI, meaning the bird's vocal energy decreased during the dry period. And we also find an interesting result. There was a significant interaction between drought and vegetation capacity. When drought happened, sites with more complex vegetation actually showed a drop in ADI, meaning the variety of sound frequency went down, fewer types of chords or pitch ranges. And at the same time, AEI went up, which tells us the frequency in the soundscape was more evenly spread. This might be because some dominate and louder species stopped calling, so everything become more balanced. So even though complex vegetation can sometimes buffer environmental stress, here it seems to change the soundscape in unexpected way, not necessary or positive. And our finding really highlights how sensitive bird vocal behavior is to climate. Among all the factors we test, temperature and humidity were the strongest and most consistent predictor of vocal activity. And we also saw the BI drop during drought, which probably means birds were calling less. This could be due to the stress, or maybe they changed their daily behavior. So for some birds, calling is linking to the breeding or defending territory. So when the environment gets tough, those behavior might be delayed or suppressed. Now, when we look at the vegetation capacity, it didn't explain much when he is only, but things change when we consider its interaction with drought. In place with high VCI, frequency diversity went down, but at the same time, the frequency became more evenly spread. So it seems that complex vegetation can both buffer and reshape how birds respond to drought in both helpful and unexpected way. And before I end my speech, I'd like to sum up a few key points. First, temperature and humidity consistent influence birds' vocal activity. This confirms that avian soundscapes are highly climate sensitive, and even small shift in microclimate can change how birds' behavior. And second, drought had a clear and strong negative effect on the BI. This likely reflects reduced vocal activity, possibly due to the physical stress of behavior suppression under dry conditions. And third, vegetation complexity didn't act alone, but when combined with drought, it significantly changed the structure of the soundscape. We observed the less frequency diversity, meaning fewer kinds of sound, but at the same time, more even frequency spread, possibly due to the disappearance of dominant vocal species. So overall, drought doesn't just make the landscape quiet. It reshapes how the soundscape is organized. So this gives us a powerful way to monitor how ecosystems respond to short-term stress, especially in fragment and mosaic environment. And so sorry, I forget put Alison picture in the slide. So anyone want to see it can find me later. And it is our reference of this research. Thank you for everyone.
Okay, bit of a change of direction now. I'm going to talk a bit about how exploding stars are helping us measure soil moisture across Australia. I'm wondering if my first slide's gone missing, but we'll see. Oh no. So this talks about the Cosmos network. It's a network of soil moisture sensors that we have distributed across Australia. It was first established back in 2010 and has since been funded and continued to be run through Tern, for which we're very grateful. So to kick things off, what are cosmic rays? Well, they're produced by the blast waves from exploding stars or supernova, or at least we think they are. So basically we say we think they are because it needs to be something incredibly powerful that energises these particles, and the only thing that we can think of that science can explain is these supernova. So the cosmic rays themselves are pieces of atoms that have been accelerated and energised, and they travel at close to the speed of light through space, and eventually some of these will reach the Earth's atmosphere. And if they've got enough energy, then these cosmic rays will pass through the Earth's magnetic field and enter into the atmosphere. So once they enter into the atmosphere, then there's the high energy cosmic rays collide with different atmospheric nuclei, and that creates these cascades of secondary cosmic rays, which we're trying to illustrate with the figure on the right here. And that's a process known as spallation. But the important thing that happens is that for each collision, the energy of these cosmic rays is diminished. So we go from high energy neutrons getting through the atmosphere to then fast neutrons and then thermal neutrons. And the important thing for us is that hydrogen is the most effective element in reducing neutron energy. And that's important because the largest source of near-surface hydrogen is water stored in the soil. So there are other sources of hydrogen, such as biomass, but these signals tend to be more fixed in time. So the signal that varies is the soil moisture signal. So how do the sensors work? Well, it counts the number of fast neutrons. And just to remind you, we go from high energy neutrons to fast neutrons to thermal neutrons. So the more hydrogen or water there is in the soil, then the less fast neutrons we're going to measure. So it's an inverse relationship. And that relationship between soil moisture and fast neutron count is defined by a field calibration procedure, which takes about half a day. Here's the sort of standard system that we've been deploying through the Cosmos network. It's got a satellite link. We usually measure rainfall with it, pressure, temperature, humidity, all important for correction procedures. Solar panel, so it's a very low power use system. And then the magic happens in the tube on the side there. There's a tube full of a particular type of gas which is sensitive to cosmic rays passing through, and then there's a counter at the bottom. So we're literally counting the number of cosmic rays that pass through this tube. More recently, we've tended to attach these tubes on their own to other stations, such as the super sites. So we're trying to piggyback on some of the existing measurement infrastructure. So what's different about cosmic ray soil moisture measurement? Well, probably the most important thing is the area over what it measures. So if you think of a traditional soil moisture probe, like a TDR probe, you probably get a measurement footprint with a circle about 10 centimetres wide. And at that scale, there's lots of variability. But what happens when you use cosmic rays is you get a measurement footprint with a diameter of about 320 metres. So that's an area of about 8 hectares. And the interesting thing about that is it gets you above that small scale variability, and it's a much better scale match with things like flux stations and for calibrating satellite imagery and the likes. The interesting thing about the measurement footprint is it varies with depth, with soil moisture. So a bit like satellite soil moisture imagery it does as well. So when the soil's wet, we see about 10 centimetres into the soil, and then when it's dry, we see up to about 30 centimetres. But the important thing to note is that the contribution diminishes exponentially as you go down in the soil profile. So really, we like to think of it as a surface soil moisture measure. So this is the existing network, and the web page is shown at the top there. We've got 27 sites currently on the web page, with 3 coming soon once they're calibrated. 6 of these are now associated with Tern super sites. So as I say, we're trying to sort of maximise the benefit of co-locating instruments. And each of these sites has a local manager, so whether they be from university, landholders, state government or CSIRO staff. All of them give us hourly soil moisture data and rainfall and a couple of other temperature humidity parameters. Older sites have now been in since 2010, so we've got a really nice record. And of course, along with all the other Tern sites, we're all about open access data. So just quickly, of course, there's a fairly complex data pipeline in the background a few people have helped us with. That involves pulling in signals from satellites and from mobile telemetry and the likes, pulling them all together, applying corrections, processing the data, and then exposing them to the outside world through our data portal. And that's got so both the data visualization at different levels of processing and the actual delivery of the data files itself. Just some examples of the data. These are 4 of our longest running sites. Daly is up near Darwin, Weenie Creek out near Charters Towers, Robson, one of the long-term super sites, and Nangara or Gingin is another super site down the bottom there. So you can just see the types of data that we've got from these systems now, long-term data series that are really useful for going back and calibrating satellites and the like. So from a network maintenance sort of perspective, over the last 15 years, we've had all sorts of challenges, from floods to wildlife filling up rain gauges like this, through to the fires in 2020 where we lost a couple of the sensors. Those of you who had any equipment at Tumbarumba might have lost it to the overzealous bulldoze operators that worked in that area recently. We've had a couple lost through lightning strikes, and then we've had some that have gone from being a forest site to a cleared site. So that creates all sorts of challenges. So it's interesting to look at how the data is being used in the literature. So we did a bit of a survey to see where Cosmos data in particular is being used. I think we're up to 140 different papers using the data now for various reasons. Most of it so far has been about technique development. So that's the blue bars that you see. But we're starting to see an increase now in the utilisation, so that's the red bars. And of that utilisation, it's mostly being used for model calibration and validation and satellites, being used in some cases for data assimilation, so to keep models on track within sensible bounds, and a little bit of water balance and soil hydraulic property type of work. So knowing this sort of information is not only good to know that people are using it, but it's also good for targeting data delivery to those markets. So the other thing I did was I did this analysis for a global Cosmos workshop that we were at recently. and we were interested in international networks of where the data is being used. So what we did is we set up a Google Scholar profile for Dr. Ray Cosmic, who now has 731 publications and 28,000 citations and a H index of 85. So he's doing fairly well, but it's just a good way to automatically track where your data is being used and how, where you're making your impacts. In terms of some of the emerging science we're involved in as well, we've taken some of these static sensors and we've bundled them into the back of a trailer, so it's 16 tubes, 16 of these static tubes, put them in a trailer, and then we've been taking this trailer out and driving through the landscape and getting some maps of soil moisture through time. And this is interesting because you can start mapping soil moisture at the scale of satellite or model calibrations. There's other work on this going on in Europe. There's German groups are using them on trains, and there's some people using them on blimps as well. So it's a good way to get spatial pictures of soil moisture. The other thing that we've been involved in more recently is looking at intensity corrections. Now, intensity corrections are about removing background signals in incoming cosmic rays. So as cosmic rays are coming in from space, we have interactions with our own sun that through solar flares and that change the intensity of these cosmic rays. So we need to be able to remove that signal to be able to get a good clean soil moisture signal. We typically do that with what's called neutron monitors, and they measure very high energy neutrons, but the problem with them is we don't know if they relate exactly to the types of sensors that we measure, which measure the fast neutrons. So what we've done is we've put all these tubes on a pontoon out on a lake near Townsville, and the idea being that by having completely saturated hydrogen signal in the background there, the only source of variation that you measure above the water is the change in incoming intensity. So we want to use this pontoon as a correction point for the Cosmos network to be able to get much better soil moisture estimates. We're also involved in the global Cosmos community. So this is a network collaboration across the world. So there's networks now, China, India, South Africa, you name it. And we're talking about hundreds of sensors out there now. So we're trying to bring everyone together and to be undertaking consistent processing of data and also setting basic standards for how people do things. Again, it's all about open access data. The idea being that once we've got this easy access of data, more people are going to use it. You can imagine being somebody who's interested in soil moisture calibration for satellites, having access to hundreds of points around the globe in one easy to access location. That would be great. So it's all about increasing data use and making our networks more useful. So just in terms of wrapping up, there was a comment at a conference I was at that sort of sums it up nicely. I think it's amazing to think that a star that exploded millions of years ago and millions of kilometres away is telling us about soil moisture today. And I'll just leave you with that image there. That was down in Hobart a couple of weeks ago. That's the Aurora Australis, and that's one of the few times that you actually get to see cosmic rays, because that's cosmic rays interacting with oxygen and nitrogen in the Earth's atmosphere to form those amazing patterns. So I'll park it there.
Okay, so good afternoon, everyone. I'm from the ANU, and I'll be talking about a project I've been leading in relation to mapping ecosystem integrity nationally using a flexible best on offer reference approach. And I'd like to acknowledge the assistance of my colleagues, Albert Van Dyke and Phil Gibbons, for their support. Thanks. So I guess the first thing is, what do we mean by ecosystem integrity? Because this is a term that has a lot of different definitions and related terms such as condition, health and quality. But for our purposes, we're defining it as the intactness of self-organising biotic and abiotic components of ecosystems and expressing this relative to a desired reference state. And that latter clause is particularly important, and we'll get to exactly what that reference state might look like. Being able to map ecosystem integrity nationally would be useful for several reasons, some of which are outlined on the screen here. And remote sensing can provide a lot of information on diverse indicators of ecosystem integrity. And one of the objectives of this study has been to determine which of these are most useful. Of course, there are also several state and national approaches to do this sort of thing, and the Habitat Condition Assessment System from CSIRO is probably the best known example of that. And our approach shares some similarities, but also important differences with approaches like HCAS. To some degree, our study has been framed by the requirements of the Australia's Environment Report, which reports annually on the change in the condition of Australia's environment using indicators like leaf area index. And what we're trying to do is essentially to untangle the natural variation in indicators like LAI from the unnatural variation to give us some deeper insight into ecosystem degradation in Australia. So in our definition of integrity, the word relative is doing a lot of work. In a very simple mathematical sense, we can quantify ecosystem integrity as the departure between the observed state of an ecosystem for any given indicator and a reference state, or what we expect that ecosystem to look like. The difficulty, of course, is defining that reference state and then also quantifying that reference state. So this is a framework from Megan McNally and others who suggests that we can define the reference state as possibly being a pre-human condition of an ecosystem, or in an Australian context, maybe a pre-European condition. Perhaps you can look at natural or pristine areas to define the reference state, areas in national parks and so forth. But for the purposes of this study, we've taken a best on offer approach to defining the reference state. And I suggest that this has two key advantages. One, it gives us more training data for informing our models, which is good. And the other thing is, I think it's a bit more application ready, because a lot of the potential applications of this sort of research relate to things like restoration targets. And if we can provide data or provide products that are more grounded in contemporary ecological realities, and I think that's quite useful. I'll briefly show you kind of what we've done and then take a step back and walk you through how we've done it. What we do is we take a given indicator of ecosystem integrity. So this is a forest height product derived from these authors using JEDI spaceborne LiDAR data. And what we do is we predict what best on offer forest height could look like or should look like across the country. And by looking at the departure between these two things, or the difference between these two things, we get this map of change in forest height, or delta forest height, a departure, if you like, which is a bit more insightful than simply the observed forest height, because we know in Australia that many ecosystems used to have trees but don't have trees anymore, and also many ecosystems are naturally treeless. Of course, forest height is just one indicator of ecosystem integrity that we can obtain using remote sensing. We took an inventory of products that are available out there that had to meet a few different criteria. So for instance, they had to be national products, reasonable resolution, reasonably accurate, and I think most importantly had to have some kind of conceptual link to the term of ecosystem integrity via a conceptual framework such as this one. So these authors suggest that integrity is comprised of several components, including things like energy capture. We can indicate energy capture through remote sensing products like GPP. And then you can also look at things like energy storage, heterogeneity, water flows, etc. And for most of these components, we can find a handful of remote sensing indicators. Overall, we found 44 remote sensing products that we evaluated in our study. And in doing so, as you can imagine, we relied heavily on the hard work of other people. And so I'd just like to broadly acknowledge the amazing data repositories that are out there and that have been really instrumental to this study. I'll cite some of these products and studies as mentioned, but a lot of them will go unmentioned and therefore uncited. In relation to a method, the first thing that we had to do was identify areas that we can use to describe what best on offer examples of ecosystems look like. So ideally, we would train models of the reference state just on observations from national parks, but as we know, national parks don't necessarily represent all ecosystems particularly well in this country. So what we did is we collated land use and land cover information and expressed it as the confidence to which we can say that these areas represent the highest integrity examples of ecosystems. And the point being that, yes, we'll train our models on places like national parks when that's possible, but when we need to start invoking information from lower confidence areas like travelling stock reserves, then we do so. And to determine how or when we have to use that lower confidence information, first we had to generate a classification of ecosystems across Australia, because best on offer and ecosystem integrity are things that are generally positioned in relation to an ecosystem classification. We did this using a K-means classification of important variables driving plant species composition according to this study from CSIRO. And then what we did is we allocated if you like, a random seed of reference sites within each ecosystem and assessed how well those reference sites represented the overall environmental domains of the ecosystem. And we did that by comparing the convex hulls of our reference sites to the convex hull of the overall ecosystem in a four-dimensional principal component environmental space. So in this instance here, if the convex hull of the reference site was significantly smaller than the overall environmental coverage of the ecosystem, then we can start to expand our reference sites to incorporate further environmental information and using those lower confidence classes as required, until we arrive at a point where we're reasonably confident that our model is going to be reasonably robust. And the key point here is that we're only adding the reference sites from these lower confidence areas like the travelling stock reserves when we can't find that equivalent environmental information in places like national parks. What we arrive at is roughly 200,000 reference sites across Australia. And at each of these, we sampled roughly 60 environmental predictors that we think control things like forest height, which we know is controlled by variables such as climate and soil and topographic wetness. From there, we used gradient boosted regression trees to train models for each of our 44 remote sensing indicators based on those predictive variables. And I guess the important thing here is because our training data only represent best on offer areas, the spatial predictions that result from these models therefore relate to a predicted best on offer state. And that's how we arrived at that map that you see in the centre of the image there, from which we can calculate a departure, in this case for forest height. Just looking a little more closely at a part of the world that I'm from, with this sort of a product, what we see is that the greatest absolute loss of forest height in this instance has occurred in these high productivity coastal environments that have been cleared. No loss in protected areas and national parks, maybe a few model residuals. And then also if you look at deserts or grasslands, again, these are areas that are naturally quite devoid of trees anyway, so we don't see a substantial loss of forest height in this instance. An important consideration that we addressed is, well, what makes a good ecosystem integrity indicator? One criterion, I think, is that it has to be predictable using our environmental predictors. So NDVI is a good example of a remote sensing indicator that is easy to predict using variables like climate and soil and topography. On the other hand, below ground biomass, which we investigated, is much harder to predict using those environmental variables. So if we're thinking about generating a reference surface of what below ground biomass should look like across the country, that's going to have a lot more uncertainty than our predicted best on offer NDVI surface. The second consideration was that our departure indicators, so the loss of forest height or the loss of biomass, these had to be related to some degree to site-based measurements of ecosystem integrity that had been measured in the field. What we used were site data collected in New South Wales that describe vegetation attributes expressed relative to best on offer benchmarks that have been empirically developed by the New South Wales government. And these site data are used in applications such as biodiversity offsetting and restoration planning, which are the sorts of applications that I hope our products might be useful for as well. So take, for example, on the x-axis here, you can see some of those site data scores. The shrub richness score, which you can see there, is essentially developed by people going out into the field, measuring the number of different shrub species in a 20 by 20 metre plot, and then expressing that relative to a benchmark. Overall, what we find is that the highest correlations to the site data were achieved with the loss of forest height, the loss of above ground biomass, and the loss of woody vegetation cover. And just to finish up on a bit of a sanity check, if we have a look at those three key attributes, the loss of forest height, biomass and vegetation cover, and their distribution across land use categories, we see that in protected areas and production native forests, very little departure in those attributes. They are essentially as we predict they should be. Plantation forest is a tricky story. But in grazing native vegetation and intensive uses where you have things like suburban gardens, you see a moderate loss of these attributes. And I think quite unsurprisingly, the greatest loss of these attributes has occurred in things like cropping areas and irrigated agriculture. I'll leave it there and happy to respond to any questions later.
Coastal wetlands, mangroves and salt marsh are important yet vulnerable ecosystems. They capture and store large amounts of blue carbon, support biodiversity, and provide natural coastal protection to the communities they coexist with. Occupying low coastal elevations, sea level rise presents a risk to these valuable ecosystems. Monitoring contemporary rates of surface elevation change using surface elevation tables, also known as ROD surface elevation tables, allows us to assess the vulnerability of coastal wetlands to sea level rise as multiple processes contribute to the losses or gains in elevation. A coordinated approach is needed for regional and global networks of surface elevation tables, and Tern, Australia's Ecosystem Observatory and member of the Global Ecosystem Research Infrastructure, GERI, is enabling the formation of the OzSET network, Australia's approach to standardising and strategically expanding coastal wetland monitoring into being recognised as national research infrastructure, with local, continent-wide, regional and global applications. I acknowledge the work of the many international coastal wetland researchers and surface elevation practitioners whose work I will draw upon today. So, the global distributions of mangroves and salt marsh have recently been mapped using remote sensing products, revealing over 52,000 square kilometres of salt marsh and over 145,000 square kilometres of mangroves. Existing mangroves and salt marsh need to adjust to sea level rise or we risk losing them, but they can and have adapted despite changes in sea levels over the millennium. The response may be that they keep pace via sediment accretion and root growth, so moving vertically up, or by moving landward if space permits. But inundation beyond their physiological tolerances will lead to coastal wetland loss, so understanding the surface elevation is critical in determining the wetland vulnerability to sea level rise. So surface elevation tables are a method that were developed by Don Cahoon and colleagues of the US Geological Survey, and they very precisely measure the surface elevation change relative to a benchmark. When paired with marker horizons that delineate surface processes and measure accretion rates, we can calculate the shallow subsidence. They're low cost, making them more accessible to a wider range of countries, yet they're high precision, so they can be used to fill a critical data gap in quantifying the shallow surface and subsurface processes that can be paired to larger spatial data sets and modelling. Webb and colleagues in 2013 outlined the utility of the method, identifying it as the global standard for monitoring vulnerability of coastal wetlands to sea level rise. They did a global first-pass assessment identifying vulnerable coastal wetlands while highlighting the large geographic gaps in data and the need for a coordinated expansion of monitoring efforts to better understand coastal wetland adaptation under climate change. So Darwin Harbour in the Northern Territory of Australia is a drowned river valley and is home to over 20,000 hectares of diverse mangroves in near pristine condition that provide valuable ecosystem services, including the storage equivalent to over 20 million tonnes of carbon dioxide in their biomass and the top 1 metre of sediment. So surface elevation tables have been used since 2016, and they're paired with water level data, LiDAR, and radiometric dating. We can see that increases in elevation in Darwin Harbour occurs largely through sedimentation in the seaward zone, and below ground contributions are limited. This tight coupling between the sedimentation in the lowest elevation zone and limited sediment increasing the importance of below ground processes in the landward forest highlights that climatic drivers are important for the health of the mangrove forests, as sediment is suspended by monsoon-driven waves in the Northern Territory, and rainfall contributes to below-ground productivity. At a continent scale, there are over 300 surface elevation tables around Australia, some with over 2 decades of data. Synthesis of this long-term data, covering a range of hydrological and geomorphic settings, allows for the identification of important biological and environmental controls, identifying that accretion enhances shallow subsidence and that elevation gains are lagging in comparison to regional sea level rise. While it also acknowledged the large geographic gaps, there's also a useful application of this data to develop a dynamic coastal elevation model to help with coastal adaptation planning. Surface elevation tables have usually been used in coastal wetlands, but now they're also being used in adjoining ecosystems, so the supratidal coastal forests, mangrove salt marsh, and even in subtidal seagrass. Taking a regional look at the utilisation of this research infrastructure, the Indo-Pacific region is currently experiencing some of the highest rates of relative sea level rise in the world. Lovelock and colleagues in 2015 identified that 69% of mangrove sites were experiencing an elevation deficit relative to the regional sea level rise, and modelled that it would be likely they would be submerged by 2070. They highlighted the importance of sediment availability for the ongoing persistence of these ecosystems. In a global assessment in 2023, Santiland and colleagues used multiple lines of evidence, including data from over 600 surface elevation tables and the paleostratigraphic record, to draw conclusions about the persistence of coastal vegetated wetlands under various sea level and warming scenarios. They identified a general 7 mm per year limit of regional sea level rise, resulting in a high probability of losing these coastal wetlands under a 3-degree warming scenario. So this was a broad sweep of the utility of surface elevation data at various scales, from understanding processes to future models and management and policy implications for the long-term persistence of these high-value ecosystems. In Australia, the network of over 300 surface elevation tables with its standard protocols and fair data practice is national research infrastructure and is part of the Australian Ecosystem Observatory run by Tern, with funding from the Australian Government. Tern is part of GERI, and hence all systematically collected long-term data from the Australian OzSET project becomes part of the harmonised GERI data assets available for global research collaboration and data integration with other coastal datasets, such as remote sensing. Tern's website has more information on the Australian OzSET project, so please use our QR code here to find us.
Okay, hi everyone. I'm going to talk a little bit about how we're boosting ecosystem monitoring in WA's climate-sensitive landscapes. Although climate-sensitive is probably perhaps not... Sorry, I'm a bit of a walker, I'll try and stay still. Climate resilient is perhaps a better term to use. Now let me just see if I can get this right. Okay, here we are. And so we've been doing this with a number of different instrumentation items, which we've sort of added to our existing super sites to help boost our monitoring capacities in these ecosystems. And so I'd also like to acknowledge that the University of West Australia is situated on Noongar lands, and the Noongar people remain the spiritual and cultural custodians of these lands. And so what is happening with climate in Western Australia? And so like much of the rest of the sort of southern side of continental Australia, we're seeing here we go. So here we're looking mainly at the southwest pocket of Western Australia, and we're looking at the annual maximum temperature anomaly from 1910 through to 2024. We can see it's getting much hotter, particularly in the recent sort of since the early 2000s to present. Correspondingly, on the right-hand side, we're looking at average annual the rainfall anomaly from the same period, and we've basically been seeing a drying trend, which started to really initiate in the 1960s, but then from 2000 has started to accelerate. And so unlike much of the eastern states, which in recent few years has had a bit of a reprieve from long-term drying with the strong La Nina events, we didn't really see any of that in Western Australia, and so our ecosystems are really facing this hot, dry trend. And there are already some pretty dire consequences of this that we're starting to see in real time. And so this occurred, there was sort of, back, okay, that one. This big sort of forest dieback event happened in the summer of 2023 and 2024, where our iconic jarrah forest species, particularly that are all around the Margaret River wine region in a very highly touristed area in the summertime, were all dying back. And we also saw that in our Banksia woodland ecosystems as well. And so some of us did a bit of a snapshot of what happened looking at the Jinjin Observatory, the Tern Jinjin Observatory, and then scaled up along a north-south transect, looking at the Banksias across the region. And so, we basically showed that, major limitation to evapotranspiration in these Banksia woodland ecosystems. At our Jinjin site in particular, we didn't actually see a deficit to the carbon cycle at that point, but there were areas further north where the heat and the dryness was even more severe, and we started to see dieback occur, and so that's where we were seeing nice, beautiful Banksia ecosystems like this turning into these crispy, dry, nothing alive systems. And so, what are we then doing to boost our ecosystem monitoring in WA? So there's been a sort of a phase one of this project, which was funded by the co-investment from the WA state government, which really looked at our sort of, I keep getting these buttons wrong, our 4 sites here in Western Australia. So we've got 3 sort of native ecosystems and a farming system. Our first order of business as part of this boosting of monitoring was to install some biodiversity transects with the help of Ben Sparrow's team at the University of Adelaide. And these transects were basically centred around our flux towers. And so we can see that in the Great Western Woodlands, we have our flux tower in the middle, and we're scaling between the mulga ecosystems that are here in the north down into the salmon gums that extend down into this sort of higher rainfall zone. Likewise, we have a transition in rainfall with our eucalyptus wandoo, and then a transition, particularly in temperature, with our Banksia ecosystems. And at these biodiversity transects, we're monitoring things like the species abundance. There's some ant samples and then some soil samples that we can then use for some metagenomic analysis as well. We're looking at some changes in canopy light dynamics and quantifying that in more detail. And this is using a whole bunch of PAR nodes that have been installed at each of our Tern super sites, so each of the Jinjin, Boyjan and Great Western Woodland sites. And this is giving us a better understanding of how light is transferring through the canopy so that we can get a better handle on the photosynthetically absorbed radiation. We're also pairing this with some measurements of canopy structural change, and to do this we're using a leaf scanner. So it's a fixed terrestrial laser scanner that essentially collects a ground scan and some canopy scans each night, so we can stitch together in really great detail how the structure in the canopy is changing over time. And I'll get to some of examples of that in a second. And then we're also boosting our sensing capacity through adding some hyperspectral sensing techniques. And so this is looking at installing some SIF and hyperspectral sensors on our flux towers so that we can capture not only the carbon and the water fluxes, but pair that with some of the remote sensing indices, but from a proximal sensing capacity rather than satellite. And so there's a whole bunch of new data streams that we now have coming through the line from this part one of this project. And so all of our flux sites have been running for many years. Our newest one, Boyjan, now has 8 years of flux data, all available on the Tern website. But we've now got about 3 years or so of leaf scanner data, the spatial PAR data within that sort of, they're about in a 1 hectare array around the flux tower. 3 years of biodiversity transect plots, so at each of those biodiversity markers there is a 1 hectare Tern plot, and we've gone back and resampled those plots every 3 years since they were established, so we've got some good data coming through there now. And also some SIF and hyperspectral sensing data for Boyagen. These sensors have been my problem children, but they're now behaving, and I'm starting to get some good data coming through from those sensors. And so essentially what we're starting to see, come on, there we go. So here we're looking at a compilation of the photosynthetic active radiation that is happening at the Great Western Woodlands site. And we can see, so this is a fairly sparse site, there's actually not a lot of trees around. And so we can see how the canopy light dynamics are changing through the canopy with the incoming photosynthetic radiation above the canopy in green, and then the upwelling component in the pale green below it. And then as well as, what's being reflected up off the ground surface. We're also able to track with our leaf scanner changes in foliage area volume density, so we can see at what height in the canopy we're seeing a change occurring in our plant area volume, which is largely driven by the leaves, as well as start to track over time. And I have to thank Darius Colviner, who's in the crowd, for producing this image. And so here we're looking at a time series of changes in plant area index from the Boyagen site. And so very quickly, because I had an 8-minute talk and I of course knew that I would waffle on, essentially we're now entering phase 2 of our Tern project. And so this is essentially looking to add a Jarrah Forest Observatory. So we will be adding a new flux tower and a new biodiversity transect so that we can start to capture this unmeasured site in the Tern network. We'll also be adding some more agro-ecosystem observatories. We've currently got one flux tower at the moment, but we're planning to add another one in the sort of drier agricultural regions near Merredin. And then expanding our critical zone observatories as well. And so that will be, come on, there we go. Adding some below ground monitoring at Jin Jin to sort of get down to groundwater. Luckily as well, we've also had co-investment for a data analyst. So one of our biggest problems is that we've got all this lovely data, but it's hard to get it actually available and quality checked and quality assured to all of you as end users. And so having this data specialist now on board will be a really great boost in the amount of data that can be coming out of Western Australia. And so with that, I'd like to thank, in particular, the Western Australian State Government, who have invested quite a lot in Tern infrastructure and monitoring capabilities in WA. And we can then, hopefully with their investment and continued work from Tern and the likes, start to really fill some of those data gaps that are in Western Australia. And thank you very much for your time. There we go.
Hello everyone. Today I'll be talking about automated on-farm biodiversity assessment using combined remote technologies. This research was part of a broader project looking at carbon sequestration and biodiversity co-benefits in grey systems in the Brigalow Belt in Queensland. And really the main driver for automating biodiversity assessment as much as possible is that standard surveys are quite costly and time-consuming, with most biodiversity assessments involving a comprehensive vegetation survey, fauna survey, and measurements of ecological condition. So the question we asked is, what if farmers could monitor their own biodiversity by combining existing technologies? And can we provide the tools to reduce the assessment load on farmers and also encourage the uptake for biodiversity assessment and monitoring at scale? So where we started was in that broader project in the Brigalow Belt. We're looking at whether we could combine satellite data with sound data to measure ecological condition. So that is, can we predict biocondition scores, biocondition being an ecological condition metric in Queensland without actually going out and physically surveying sites. Biocondition is like a lot of condition models, it's fairly representative. You use a suite of site-based and landscape-level vegetation attributes. Measured at any given location, these are scored relative to benchmarks to give a condition score, which is representative of biodiversity value for that location. So the model involved collecting bird data, so this was bird presence absence data from passive acoustic monitoring, combined with vegetation indices and patch metrics from satellite platforms. And the model performed reasonably well. The modelled condition scores on average were plus or minus 8% compared to standard biocondition scores. I think the R-squared was just over 0.8 for the model. So we were already using automated multi-species bird recognisers for acoustic data for that previous project. We were using BirdNet, so this is commonly used. People doing acoustic research. BirdNet is a machine learning approach developed by Cornell University as a massive library of bird calls, including Australian bird species. However, in the last few months, SpeciesNet has become available as open source machine learning software for camera trap images. So this was originally developed by Google, and so we've incorporated that into the monitoring tool. Those two images at the bottom are examples of the SpeciesNet output. So the way we brought it together was we combined data from passive acoustic monitoring, from camera traps and from satellite platforms. The data was processed using the existing machine learning models, so this was BirdNet and SpeciesNet. We combined it with the ecological condition model we developed in Brigalow, and some fairly standard spatial analyses using GIS. We further analysed and integrated that data using custom Python scripts developed at QUT. And then the outputs for farmers were really ecological integrity scores and classes and farm reports informing management and ongoing biodiversity assessment and monitoring. So I guess the best illustration is to look at the prototype itself. This is conceived as a modular tool. So at the moment we were focusing on 4 major indices, one for camera traps, one for acoustic recorders, one for the ecological condition metric, and one for landscape. So each of these indices was underpinned by multiple metrics, and these were combined using a weighted sum approach. And at a survey point level and at a site level, so at a farm scale, these indices were given ecological integrity scores based on predetermined thresholds and weights. So this is the tool, well, part of the output. So this is an interactive map. Each of those green points is a survey location. The central red location is all of the survey points aggregated together. To look at one specific site, so for that each location we have an overview image. We have a camera trap index, so this is underpinned by multiple camera trap metrics. These are aggregated and normalised, giving a standard, like a standardised camera trap index score between 0 and 100. It's given an associated ecological integrity class for that index. Part of the camera trap output is we provide a visualisation of the relative abundance of the main species being detected. These are native and non-native species. And then for each index itself, we give, I guess, a summary of the camera trap index and each of the individual metrics that are make up that index. For camera traps, we give some visualisations of the main species being detected based on relative abundance. So we have native and non-native species. Similarly, for the acoustic recorder index, we have an index normalised between 0 and 100 and an integrity class made up of multiple metrics, including relative abundance of the main bird species being detected by the acoustic recorders. And once again, we provide this acoustic index plus the multiple metrics that make up that index. For the acoustics, we provide a visualisation in the form of the acoustic spectrogram. So this is a 10-minute snippet of dawn chorus for that particular location. And we have an associated audio sample of that spectrogram giving some indication of the bird species found at that location and a broader, I guess, summary of the soundscape. The third index used in the tool is the ecological condition. This is from the model, not from a standard survey. And once again, this is already normalised, so it's assigned an ecological integrity class. The final index we've used is a landscape metric. So this is made up of fairly common fragmentation indices combined, but measured at multiple scales to give an overall index and integrity class. Typically, these score very poorly in these landscapes just because they're highly fragmented. And this kind of informs the final component where we combine them all together. So we actually just combine the non-landscape indices together at first to give a combined score and an integrity class, and then we add in landscape to provide a combined landscape and non-landscape measurement for that particular survey point. So every survey point gets this summary data assigned to them. The central red location, as I said, is a summary of all the survey points. So this is aggregated across all the survey points within the boundaries of that farm. Once again, we split out the landscape and non-landscape metrics indices at first, then we add them in to give a combined index at a site or property scale. So the deployment and analysis workflow, this is really conceived as farmers collecting data and then, deploying equipment and then the data being analysed by specialists post-survey. So really the workflow is that we have a discussion with farmers where we would like to deploy recorders and camera traps. They might have some idea about where they want to target assessment on their property. We package everything up in a box, including a dedicated tablet with mapping and instructions. We send it out to them. They go and deploy their equipment. Following six weeks of deployment, they'll put it all back in the box, send it back to us, and then we will analyse it in the lab and provide reporting and recommendations based on the outputs. So the next steps in this project is we're currently testing it in the Brisbane Valley with healthy land and water across multiple grazed properties, but we're also looking at testing it across the old tar sites that Dave Rowlings discussed earlier in the day. We're also looking at expanding and testing the ecological condition model in additional bioregions and developing regional thresholds and benchmarking based on literature, external data sets and expert opinion. And we're really interested in farmer engagement with the technology. So what do they find difficult? What do they find easy? And how can that inform the further development of the tool? So I'd like to acknowledge Meat and Livestock Australia and Healthy Land and Water for funding these projects and all the participating landowners for their contributions. Thank you.
Thanks. Hi, everyone. I'm Paul Ray. I want to give a bit of an update on the Acoustic Observatory, and that is quite nice after what David's been talking about. So this is sort of joint work with folk at QUT and with JCU, where I'm now based. I want to start by acknowledging the traditional owners of the land here, the Turrbal and Yugara people, as well as the traditional owners of the land where we collect data for the Acoustic Observatory. And to acknowledge that the traditional custodians have always played an important role in preserving and looking after land and sea country. So the Acoustic Observatory started as a result of a LEAF grant, which we got so about 5 years ago now. It was a joint project with people from other universities, and it was about continental scale biodiversity monitoring. In particular, we were interested in sampling biodiversity across ecoregions across the nation. It was very much and unassumedly a big data first approach, so quite a different way, particularly at the time of doing kind of monitoring. We were collecting data. We didn't have particular questions other than just general biodiversity monitoring, which we were trying to answer. We've got deployed, I think, about 365 sensors now, but the goal was to have 400 sensors deployed across the continent. They were continuously recording, and the data is being made available on an open access basis, so it's sort of fair data and designed to sort of support research in computer science for creative use as well as sort of ecology. So the idea of the observatory is that it's an observatory, so it's a set of data that is available for different uses, just like an astronomical observatory. It's not a curated data set. It's not a data set that has any sort of specific kind of purpose other than general biodiversity monitoring. So it was really a new way to understand what's going on across sort of our nation. The deployment started around 2019. We had a few hiccups with sort of COVID and with bushfires and things like that. We sort of co-designed some recorders, these Frontier Labs bar solar recorders with which we used. The sort of LEAF grants is an infrastructure grant. They don't sort of fund operations. So the actual operations, actual collecting of the data, of the SD cards and things like that really runs on goodwill. It runs on goodwill of the partners we have with, I think we've got over sort of 50 partners with national parks, with rangers, with NGOs, BirdLife, and other landholders. That sort of worked well. We've got the data kind of rolling in from across many sites. We're up to, I think, about what we've got, the 365 years of data. So the data is accumulating a little bit slower than we originally hoped, and we've got sort of about 300, more than 300 users, and that's without really advertising what we can do with the observatory. And I'm going to talk a bit about what we're now able to do with the observatory. The observatory is sort of underpinned by a data pipeline, which is a project with the ARDC, also supported by Google. The data pipeline does the analysis. In fact, it's more than just the analysis and storage and management of the data. We also run sort of workshops and other activities and sort of training exercises around how to actually sort of collect, analyse ecoacoustic data. It involves sort of Tern, the Atlas of Living Australia, and other ARDC projects. It's, as you can say, it's kind of complicated. There's a lot of moving pieces and things going on. It also supports the EcoSounds platform, which is a set of different projects collecting ecoacoustics data, and I think altogether we've got almost a petabyte of data now. In order to analyse the data, and David sort of mentioned this, is BirdNet's become very popular. The thing with BirdNet is a deep learning network, and it's trained on Northern Hemisphere birds predominantly, so it's very good for North American birds and European birds. Works okay for some Australian species, not so well for others. It doesn't work so well for some of our threatened and rare species. It also doesn't tend to work so well for species in the north of Australia. What you can do though with BirdNet and these deep learning networks is you can do a thing called sort of transfer learning. It's the idea of these sort of embeddings where once you've got a network which is sort of trained on some data, you can sort of take out some of what that system has learned. In this case, if you like, the squawks and trills and warbles of birds, and then build lightweight recognisers on top of that, or you can build search, or you can use it for sort of unsupervised learning for clustering and things like that. So by using things like kind of Xenocanto, which is this sort of open access set of bird calls that birders have collected, things like BirdNet and Perch, the Google bird classification system, have been trained on these sort of North American and European species, but we can kind of make use of that learning to be able to recognise sort of other species which we may be interested in for some of the things which we need to do. So in particular, if we want to recognise, say, threatened species, for example, to drive something like the Threatened Species Index or an Invasive Species Index, we can use those techniques I mentioned using the sort of the deep learning embeddings, sort of such as search. We can build lightweight recognizers, or we can build sort of unsupervised clustering approaches to detect sort of species. So a couple of years ago, we wrote a paper really looking at what was the potential of the Acoustic Observatory to detect threatened species. So we were just looking at maps of where threatened species might be. And then more recently, we've actually used it to actually build some recognizers and detectors to actually look for threatened species. So we've identified, I think, 76 species altogether, which we should be able to detect acoustically, some using sort of BirdNet out of the box, some using these sort of embeddings and lightweight recognizers and sort of search routines. So some sort of birds, some frogs and some mammals. We've then analysed data from the Acoustic Observatory across the sites which we have, and we've detected 43 of the 76 species. So we've validated a lot of those, so we know that they really are there, and the system has sort of worked well. What that really means is that we've been able to use the Acoustic Observatory to detect threatened species when the observatory was never actually designed a priori to do that. So we sort of validated, in a sense, the Acoustic Observatory and actually being able to use it for something. Some just examples of threatened species we've detected are brown tree creeper, blue-winged parrot, from koalas, Wallam sedge frogs, yellow-bellied gliders and other species. So we were sort of happy with that. What it essentially means is that the Acoustic Observatory can be used. We can use it to detect threatened species. We can certainly use it to detect invasive species. We can use the analysis tools, and by using and adapting some of the analysis tools, we can also detect Australian species, which the tools don't detect out of the box. And we're now in a position to actually sort of feed data from the Acoustic Observatory into things like the Threatened Species Index, into State of the Environment reporting, could be an Invasive Species Index and things like that. Thank you.
Hi everyone, I'd like to talk to you today about a deep dive into the Tern Australia Soil and Herbarium collection. So the new facility was officially opened in December 2022, and it's a purpose-built facility for the collections, which is also climate controlled and has an integrated pest management plan for South Australian biosecurity purposes. We're a CA12 accredited facility. All of our collections are available for loan, and we also have a donation and exchange program with state and national herbaria. In the collection, we have almost 200,000 plant and soil samples, and with those samples, we have a whole series of guidelines which explains how we collect the vascular plant collection, how we image the collection, mounting guidelines, overall collection management guidelines, and more recently, a rapid ant sampling protocol, and more is coming. So just an overview of our sample types. We have about 60,000 plant voucher specimens, as well as leaf tissue samples, or plant tissue samples, soil pit samples, soil subsite samples, and soil metagenomic samples. And the collection is very unique in that it includes both soil and plant collections, and we also have supplementary data that includes contextual information on site location description, site photos, including an automated stitched panorama, vegetation structure and composition. We do a full floristic inventory of our plots. We collect basal area. We also conduct a full characterisation of the soil, geomorphology and substrate. So we collect the samples across the Tern ecosystem surveillance plot and transit network. There's almost 1,000 1 hectare monitoring plots across Australia with just over 400 plot revisits. And these plots are located and representative of Australia's major vegetation groups and major biomes. So just a little bit more of a look into the plant specimens and samples. So we refer to this as our herbarium specimen collection. And there's two different types, the plant voucher specimen, which is the main specimen that's collected on a plot. And with that plant voucher specimen, we destructively sample leaf tissue from that sample. In addition to the herbarium specimen collection, we also collect dominant species. So these are used for population genetics. We recognise 4 to 5 dominant species in a plot, and we collect 4 individual samples for those. The plant voucher specimens are used for taxonomic revisions, phylogenetics, ecological research, and increasingly traits. Plant tissue samples are used for a whole bunch of analyses, including genomics, molecular, isotope, and the dominant species are collected for population genetics. The plant voucher specimens, we collect a representative specimen for each species that occurs within a 1 hectare monitoring plot. We focus on vascular plants, which are pressed on site in a day press and then transferred to a field press of an evening and dried throughout the duration of the trip. We collect fertile material if possible and enough material for a duplicate to include in our exchange program with other herbaria. All of the samples or specimens are uniquely barcoded. They are also determined at the state herbarium, and we mount specimens in archival material. They're also imaged with our imaging program with a unique label. They are frozen and then stored in a cool room at 17 degrees and relative humidity under 50%. We have an exchange program with herbaria, and this is an example of a workflow that we have with the Western Australian Herbarium. We also collect the plant tissue samples, and these are destructively sampled from each plant voucher specimen. Dominant species are collected from 4 individuals, and we place them in a synthetic tea filter bag with a unique barcode. They're stored in airtight containers with silica gel to absorb moisture. Airtight containers are stored in 17 degrees Celsius in the main collection, and the silica gel is monitored and replenished when required. This is our workflow here of the team processing the specimens, putting barcodes on the leaf tissue samples and the herbarium specimens. The soil samples, we have 3 unique sample types. Firstly, beginning with the soil pit, where we do a full soil characterisation, collect bulk density, pH and EC, and these specimens are used for physical analysis and chemical analysis. So samples are collected down the soil profile, which is located just outside the plot. The soil subsite, there's 9 subsites located across the plot to represent the variation in microhabitats, and these are used for physical and chemical analysis. At the same subsites, we collect soil metagenomics, which is the top layer of soil at each subsite. These samples can be used for biological analysis. Soil pit samples are collected down the soil profile. We collect about 500 grams of soil for each sample. The pit is dug just outside the southwest corner of the plot. When we get back to the lab, these samples are oven dried at 40 degrees Celsius for 48 hours. They're sieved and ground in the lab and stored in sealed screw top containers, which are uniquely barcoded. And they're arranged in the collection, stored at ambient temperature, arranged by plot, and they're stored in pallet racking. So the soil subsite samples are the ones that are collected across 9 locations across the plot. And it's where we dig a pit to 30 centimetres and collect samples at 10 centimetre increments. 500 grams of soil sample is collected for each increment. And just like the soil pit samples, these are oven dried at 40 degrees Celsius, and they're sieved and ground in the lab, stored in the screw top containers, uniquely barcoded. We have 27 subsite samples per monitoring plot, which are also kept at ambient temperature and contained in the collection on pallet racking. The soil metagenomic samples are also sampled from across the 9 subsites in the plot, where 200 grams of soil is taken from each subsite, and it's the top surface of the soil. These are stored in calico bags, sealed in a plastic bag containing silica gel. The silica gel is monitored and replaced when required. These are stored in a cool room at 17 degrees Celsius. We have a new collection for invertebrates, so we are rapidly collecting ant samples, and the method that we use is to attract and rapidly capture the dominant ant species. So we have 20 points within the main 1 hectare monitoring plot. We use a fish-based bait with honey. The vials are placed flush on the ground and they're left out for 15 to 30 minutes. On collection, we have doused cotton wool with ethanol. We plug the vial and then they're made available to Alan Anderson, who processes pins and barcodes the samples. The leg samples are sent to Bold for DNA extraction and sequencing. The unvouchered samples are stored in a wet collection at the Tern Australia Soil and Herbarium collection. So how do we access the Tern samples? So as Guru is or going to mention, EcoPlots has been extended to include samples. So a user can now visualise and request access to the samples. So the same sort of facet design is available in EcoPlots, and there's a new facet for sample types. So a user can actually filter on those sample types from across the whole plot network. Can also search by the map tool, and EcoPlots is available via ecoplots.tern.org.au or by scanning the QR code. The great thing about searching on samples in EcoPlots is a user can submit an expression of interest. The expression of interest is made available to Tern to assess. It also includes a summary of the samples that have been requested. As part of the process, a user can download their expression of interest, and the data download package includes the samples plus the EOI as PDF, and also as a JSON file for the Tern team to upload in their collection management system. Another great feature of Tern samples is IGSN numbers have been minted. So every sample has a landing page and an IGSN number. So here's an example of the plant voucher specimen with all of the details associated. And for those specimens that have an image, that's also included on this page. It's a 10-step process to submit a loan request for Tern samples. So a user simply goes into EcoPlots, submits an expression of interest, right through to Tern making the samples available upon agreement to the sample use and terms and conditions. Once the loan applicant has undertaken analysis, Tern are notified of the results, and the results are cited in publications, and this is all documented for future reference via a loan agreement. So we actually look forward to receiving new requests to borrow the Tern samples, and thank you very much for your time today.
Hello, my name is Andrew Tokmakoff, and this is a video presentation which I'm giving on behalf of the co-authors, which you see in the list below. The paper's Engineering Monitor and Extensible Tool for EMSA Field Data Collection. This is an 8-minute talk, so it'll be pretty fast. So let's get some context first about what's Monitor. It's a software system that facilitates field data collection according to the EMSA standards. It's a type of web app that can function offline and can also work on a multitude of devices, so tablets, phones, and desktops. So why is this important for ecological science? Well, given the fact that there's often been non-standard sampling mechanisms, data silos where people don't share, it's expensive, knowledge loss when people stop working in the space. What we're doing is we're working towards the ability for ecological data sets to be collected not by just one entity, but by many, and also for it to be shared globally, not just in Australia. So how do we go about developing this system? So we have a protocols team inside Tern, and we interface with them, and they do the ecological research to specify the modules and the associated protocols. So what sort of data is collected, and with what tools do they collect it, and what are the techniques they use. And we take that and translate it into code, which the protocols team then use, and they provide some feedback to us on the implementation to see how close to their needs we have achieved. All of our code is stored on GitLab, which is a third-party hosting provider. So also we have our issues there, and also the artifacts that come from the build process. And the CI/CD pipeline builds our Docker containers, which are pulled into the production VMs on Amazon Web Services. So that's what we use in terms of main tooling. Here's a very high-level diagram of the system. Essentially, data is modeled in the core, which is in the middle there, and then it's presented to the web app via JSON schema. And the web app uses this specification to inform what it's going to collect, and also how the UI is presented. You see there also an export function which transforms the collected data into the ABIS standard, which is then delivered to the BDR. So this is essentially a semantic web mechanism with a graph representation of the data. Here you can see our repository started roughly 5 years ago with over 15,500 commits to it, so quite a lot of effort gone in. To give you a little bit of an idea of what the web app looks like, here's a few screenshots. So here we see there's a collection context at the top, which is a project, which is organizational, and then a plot, which is spatial. In this case, it's not selected, and a visit, which is obviously temporal. So when a user logs in, so you have a user context, and then you have the actual other context that I just mentioned, then a set of protocols is enabled based upon that. Here you see the green and blue steps on the left-hand side of a workflow, which is a pattern which we employ throughout the web app for all protocols. So that's allowing steps to happen inside them. We also have access to local images and media for observations. And here you see location being collected, which is not that surprising, but if you connect an external DGPS like a Trimble through Bluetooth, then you can get pretty good accuracy on your samples. When users are expected to select from a drop-down, that set of drop-downs, we call that a categorical, and it's in alignment with the ABIS specification. Only valid options are presented, and if they're required or not, then we enforce it if it's required. Otherwise, if it's optional, we'll let it through if they don't select it. You can see protocols that are in process here, so you can operate on multiple protocols at the same time, and then you can also renew a protocol, resume a protocol if you would like to, or you can bin it if you don't want it anymore. Okay, now let's have a look at some of the technical aspects of what we've developed. So offline first is critical for us. We need to assume that the network access is either spotty or even unavailable at times. We also need to have modularity so that we can add new protocols reasonably easily and fast, not have to rebuild everything. And we also have many quality checks in the data pipeline. So it's on device when it enters the backend, and also when it leaves the backend and heads to the BDR. Also a key aspect is that we separate the org and core functions, which means that program and project management is separated from our core data collection functionalities. So let's go on to now something a bit interesting is about using agents as a tool. This is something that's exploratory. We just started to look at. So agents need access to context information in order to perform meaningful actions. So they need access to the data, and MCP is a mechanism that allows them to do that. So we've implemented an MCP server which sits in front of our core data, which allows access to that information. So that means that we can now consider new interaction paradigms with the data. So from a management and curation perspective, but also maybe into the future also for data users. So things like asking in native language about recent observations, or tasking an agent to identify long-term trends, or assisting with data quality or curation. So here's a few snapshots of what we're able to do at the moment. So we integrated into the core system, and I said, okay, please use content type discovery to tell me what's in there. And it said, oh, okay, I found all this stuff. Discovered quite a lot, starting with the key data types, and then it discovered also the different survey types that we have, and it discovered the flora and the fauna, different ones, plot and site management, and then the lookup tables, which are those categoricals I mentioned to you before, defines the different types of equipment we have, etc., etc. And a whole bunch of other custom data types like weather, the observer details, and it also summarized quite nicely. It appears to be a sophisticated ecological monitoring platform likely used for biodiversity research, environmental assessments, or conservation management. The structure suggests it follows standardized protocols for ecological data collection and management. Would you like me to explore any specific content type in more detail or help with a particular aspect of the system? So this was essentially from a blank slate, just saying, here's the data, what do you reckon? So then I said, okay, let's have a look for some recent surveys, and it identified 2 bird surveys that occurred recently, and presented some information on that as well. And this is all just with natural language. So looking ahead, what's happening? Well, we've been in production for a year, and user feedback has helped us fix up bugs and also improve the user experience. Plenty of small changes all over the code base. We find that people use many devices, more than just the ones that we recommended, and the offline first approach is doable, but it also requires a little bit of a headspace shift for our users to understand about network availability and unavailability. And having an adaptive system makes for fast changes, and we're seeing users adopt some pretty complex multi-user workflows when they've got a network and multiple people collecting at the same time. Looking forward, we want to create a web curation tool, for also some determinations tooling. We want to have data being available in a very ready, usable representation as part of the export. And of course, we want to dig a bit further into this agentic aspect. So to summarize, the system's flexible and allows a self-documenting approach, which connects the web app and the core backend. We're exploring how agents can be part of this, which is a very exciting new avenue, as we're looking at building out the tools for Australia's national long-term ecological data set. If you've got any questions, please ask. Thank you.
All right, let's see. All right, good afternoon, everyone. I'm really excited to be here. So I just started as a lecturer at the University of Newcastle, so I think my talk is going to be a little bit different today. I'm not a user of Tern data, but I've been a critical zone scientist for the last 10 years back in America, and so I've heard rumors and I've used some of the data throughout that. So really, the talk today is to present a little bit of a subsurface perspective. We've had a lot of remote sensing and how to observe the surface, and I really just want to bring a little bit about what I think about ecosystems and how they work together. And I think after sitting through most of the talks today, I feel like I'm in the right audience, and so afterwards I would love to get coffee, have a chat, and talk about this a little bit more. So the title of the talk is Uncovering the Hidden Links Between Rock and Root, and I just wanted to start with this image. Particularly with all of the remote sensing data we've had today. So this is an image of a drone flying over what I would consider to be a fairly curious landscape. This is in the northern hemisphere. It is in Georgia, but it's a hot, humid climate. But really, I want to draw your attention to the trees in the center. And I don't know if you can see them from the back of the room, but there's 4 full-grown humans right in the bottom there. So these trees in the middle, they're 15 to 20 meters tall, and they're basically growing out of rock. The other thing, I hit the wrong button, but you can see the well-established forest in the background, and you can see all the moss and lichen growing on this rock. What you can't see from this elevation is we find desert species, so you can walk around this outcrop and actually find cactus growing. So again, to me, there's a couple of questions. There's 2 of them I want to present today that I'm really interested in. But I really asked myself, like, looking from the surface, how can you have these two very different ecosystems in a similar climate existing in the same spot? And this might be my training as a geophysicist, but I think that we have to look to the subsurface. So the first question that I think about a lot is basically what makes this rock able to withstand or be in this hot, humid climate, right? What is it? Something's different about it. It's exposed at the surface when nothing else is. And then secondary to that is how does that impact ecosystem development? So now what you're looking at is a cross-section through that Earth. So thanks to many of you in the room, you've made all of your remote sense data really accessible, and I can find LiDAR point clouds and process them. So what you can see here is the ground surface. You are looking from that drone down. This little guy right here was that stand of 15 to 20 meter tall trees. And all right, I guess it's going to go. But basically the question I'm really interested in is, what does the subsurface look like here, right? Are the trees located preferentially because there's subsurface structures that allow them to thrive, or is there a feedback system where the plants and the ecosystem themselves are shaping that subsurface structure? So what I do is I use near-surface geophysics to kind of fill in that white space. So what you see on the screen now is velocity, which is how fast sound travels through the surface. So red is slow, and the dark blue colors there are fast. So this is a crystalline rock. So for really simple interpretation, red colors can hold water and blue colors cannot. And so again, you look down into the subsurface and you can kind of see the forest is actually distributed roughly where it's supposed to be, basically where it can get water. The other thing I don't have in this talk, because it's only 8 minutes, this is actually a geologic boundary. This rock over here has a slightly bit more biotite in it than anything over here. And then again, anything where this blue comes to the surface, those are those outcrops at the surface. So just to kind of explore a little bit more, I'm going to zoom in right in here to that stand of trees in the middle. We, as a field team, when we were out there, we had nicknamed it the Oasis, because if you're out there, it was really hot, humid, muggy, and the trees actually provided the only little bit of shade that we had. But what's really fascinating about this is, remember, these are 15 to 20 meter tall trees, and they were growing in maybe 10 to 15 centimeters of soil. So we had an auger. You kind of basically can kick the soil out of the way with your foot. You drill down, you got this backpack drill, and then all of a sudden it just drops, right? And so it drops into these fractures, and if you look into these fractures, you can actually find roots. So again, from a geophysics point of view, I'm always really interested in understanding what the subsurface or the bigger picture looks like. I've kind of outlined the top of that canopy from the LiDAR, so now you're not looking at the point cloud, you're just kind of looking at the first returns. And then this is ground penetrating radar underneath that stand of trees. So you can, ground penetrating radar, you can see a lot of reflections in here. So this is probably not soil, but this is probably where the rock is cracked and fractured. You can get roots and you can get water in there. It's showing up pretty good on this screen actually, but this kind of dotted line here, there's actually some amplitudes in here, so this is probably fractures that are starting to form. So again, this needs a little bit more work. We have colleagues, it's in review right now, but basically this is that complex feedback system I was talking about. Rock takes a long time to weather, but here's probably some evidence from a structural point of view where the trees are probably impacting and accelerating weathering. So basically, just to kind of roll with this, me and another colleague, before I left for this position, had put together a proposal that we had submitted to NSF, which is the ARC equivalent. And this is really why I wanted to come to this particular conference and hear about what sites you're working on, where you're working. But we had kind of put together this hypothesis or this model, very process-based, that we had called biotite and biocrusts. And it was focused in the US around these, I had called them outcrop islands, right? These weird islands of rock embedded in the forest. And there was really two questions. For this audience, these, oops, it won't let me go back, will it? Oh yeah, well. So for this audience, these ones are probably more the geology, geomorphic questions. These are related to basically why is the rock there? Right, and we had a hypothesis that rock has low fractures, so you can't initiate weathering. And then there's some other literature evidence I don't have time to go through right now, but that rock particularly is probably lacking in biotite and phosphorus. The other thing that we talked about, which I started to develop with my colleague and learn all kinds of cool stuff about soil chemistry and all kinds of fun stuff, is that basically once you have this rock, if you walk around these weird landscapes, I've been to a couple of them, as the trees die, all that stuff runs off the rock. all of that organic matter starts to build up. And so we kind of had this idea in our head that basically the plants are dying at a rate faster than the rock can weather, and you end up burying this outcrop from the bottom up. So you end up with accelerated weathering, and then we kind of hypothesize what we might see in the soil chemistry. We might see pH rise, we might see phosphorus rise, nitrogen rise. And so we looked at this as a whole system as basically a way to manage or to look at nitrogen carbon cycles in a really extreme environment. Right before I left, I went and found another one of these. So this is not the same outcrop, but you can see the extreme contrast in vegetation, subsurface structure. Me and my PhD student at the time, we went and actually collected some samples. You can see nitrate, pH, and phosphorus in the soils there. Again, this is like hot off the press. I think I got this right before I got on the plane a month or two ago. But basically, it was consistent with our hypotheses. So what I'm thinking now is I'm really interested in having coffee, learning about more of your sites. I've zeroed in, of course, on Bald Rock National Park. Every time you guys show a map, I notice that there's not a Tern mark there, but there's one pretty close, mainly because, again, this is very similar, right? You have this lady standing on this big outcrop, really dense forest, but what I'm always really attracted to is the really big contrasts. So, again, you can pull publicly available LiDAR data. Here's a cross-section across Bald Rock, and really I want to know, here you have your well-established forest. Is there something going on in the subsurface over here? Why don't we have it going on here if it's complete water control? So again, this is what I'm really interested in. Would love to have a chat and talk more. So with that, thank you.
Good morning. I'm Grant Perriott from the City of Gold Coast, presenting with Dr. Lucy Reading from QUT. We're here to share the collaborative journey that led to establishing a critical zone observatory at Springbrook. This is a science-led, multi-stakeholder initiative designed to protect ecological and hydrological integrity of one of Australia's most important natural systems, the Gondwana rainforests. It's about more than water. It's about resilience, World Heritage responsibility, and integrating people, policy, and ecosystems in the face of climate change. This work is made possible through strong partnerships, QUT, Seqwater, the Queensland Government, and Tern, who have already contributed through CSIRO's eco-hydrology team at Springbrook. Unfortunately, we have been unsuccessful so far in securing ARC linkage funding, so we're now actively pursuing new funding pathways and in-kind commitments to carry this vision forward. Next slide. At the core of this work is deep respect for Indigenous knowledge. This quote from Rosaline Best expresses the concept of clever water, that water is not just a resource, but a living presence cycling through all life. We're taking this seriously. Our monitoring frameworks are being developed with the Kombumerri people to embed First Nations perspectives, not as an add-on, but as integral to how we understand and care for country. Next slide. Local governments are increasingly responsible for safeguarding water, land, and biodiversity, but we don't have scientific reach needed to meet these complex challenges alone. That's why this collaboration with QUT, Tern, Seqwater, and state agencies matters. It combines on-ground governance with research capability, creating robust, scalable models. This isn't just a Springbrook solution. It has potential as a blueprint for regional and national integrated ecosystems and catchment monitoring. Next slide. This began from the ground up, literally. We started with no funding, just QUT student projects and a shared concern about invisible groundwater systems. That modest start grew. City of Gold Coast seed funding started this off with partnerships with Seqwater, the state, and eventually national attention through Tern. It shows what's possible when local need, scientific leadership, and institutional trust align. But now we need enduring support to keep those long-term monitoring goals going. Next slide. As a local government, we're often first to see environmental pressures, but we're not resourced to lead complex research alone. Our role has been catalytic, enabling land access, providing initial funding, and supporting real-world applications of science. This partnership lets us go beyond compliance. We're stepping into a stewardship role, and to sustain that, we need the kind of national infrastructure and support that Tern offers. Next slide. We are facing deeply interconnected, wicked problems: climate shifts, water stress, biodiversity decline, and these cannot be solved in isolation. Springbrook is the headwaters of the Hinds Dam, our drinking water source, and part of the Gondwana World Heritage Rainforest ecosystem, which is under increasing pressure. We face uncertainty in groundwater recharge, unregulated extraction, lack of long-term eco-hydrological data, and limited funding for the kind of monitoring that we need. What we're doing is weaving together diverse knowledge systems: Indigenous water law, hydrology, ecology, and applied governance. This is about creating a model of collaboration, resilience, and respect for country. And I'll now hand over to Lucy to talk about the project.
Thanks, Grant. So as Grant mentioned, we really started from the ground up. We started at Springbrook and there was no monitoring infrastructure in place, no groundwater monitoring and no surface water flow monitoring. So to start this project, we really had to try and find people that we could engage with. So we started to talk to the community, and by working with City of Gold Coast and the community, we were able to find privately owned groundwater bores that we could monitor. And this was really the start of our monitoring network. We used these bores to monitor the water levels and the water chemistry, and started to slowly build up our water monitoring network across the Springbrook Plateau. This map on the right shows the location of our groundwater and surface water monitoring sites. The red dots are the bores that we're monitoring, and the yellow dots are the surface water sites, so creeks and springs that we're monitoring. The surface water sites are on top of the plateau and also going into the valley, and all of the bores cover the plateau. By having enough partners involved in this project, we had sufficient funding to start installing some dedicated monitoring bores at Springbrook. So these bores are now able to be used as part of long-term monitoring of groundwater levels and groundwater chemistry up at Springbrook. The surface water monitoring component included in-kind support from the Queensland Government. So they set up our gauging stations at Springbrook, and we've been able to continue the monitoring of water flows at those gauging stations. This project really just keeps growing and building. So we started off with setting up that monitoring network, and then over the last 4 years, we've been able to continuously collect data on the water chemistry and water levels. And we've used all this information to make assessments of the groundwater recharge rates at Springbrook. We've looked at how these have changed spatially and temporally using multiple methods. We've also made assessments of the water chemistry and water quality. We've used these assessments to try and look at interactions between groundwater and surface water, and also look at interactions between groundwater at different depths. We've then added in eco-hydrological monitoring, so starting at one site where we were able to monitor vegetation, soil moisture and climate, and then starting to grow our eco-hydrological monitoring with partnership with Tern and CSIRO, and that's really started to happen more this year. We've been building conceptual and numerical models, so we've put all of the data that we've gathered into these models, and an example of the conceptual model is shown here on the right, where you can see different aquifer zones and interactions between the groundwater, the springs, surface water and vegetation. So we're really building all this information together, and it's something that we can just keep adding to as we collect more data. We've been developing a visualization tool with QUT's Visor team, and this is really a critical part of our communication strategy, so I'll show you some examples of that in a moment. We've been testing out some new water sampling and monitoring technologies, including a custom-built fog collector. So that's this photo on the left here. It's just been installed up at Springbrook, and it was being built at QUT, and we've been testing it out recently. We're also testing out some other smart sensing methodology, so remotely sensed monitoring of electrical conductivity, pressure, and humidity. We're testing all those out at the moment. We're testing them both at QT and in the field. And all of these methods are transferable. So we're testing them at Springbrook, but we're able to use them in other settings as well. So we can just keep growing this monitoring that we're setting up. And we're building all of this together at the moment to establish a critical zone observatory at Springbrook. So here's some examples of the visualization platform that we've built for the Springbrook project. The visualization platform starts with a video introduction from Grant and I, where we talk about the project, about the significance of the project and what we've learned. So anyone who's interested can get onto this website and start to learn a lot more about the project and the findings. But more importantly, it's an interactive tool. So it allows anyone who's interested, so community, scientists, to come and have a look at where we've been monitoring up at Springbrook and really dive into some of that data. So they can see the trends in the water levels and the water chemistry. They can see where we're carrying out our monitoring. And there's even some 360-degree videos, so you can become immersed in the environment, and some drone footage so you can explore the magnificent environment of Springbrook. So there's so many reasons why this Springbrook Critical Zone Observatory is really important. Springbrook's a biodiversity and endemism hotspot, and it includes not only the Gondwana World Heritage Rainforest, but also cloud forest. It really makes it very unique. The 2020 Conservation Outlook for the Gondwana Rainforest of Australia was significant concern. So this was shared at a UNESCO meeting overseas, and Australia really need to respond to these concerns. And we can only do that by having some monitoring data. This year, following on from that, there was a Gondwana World Heritage Climate Vulnerability Workshop, and that identified multiple stresses that are likely to impact the Gondwana rainforests. Springbrook in particular is home to many endangered and vulnerable flora and fauna species, and these are all likely to be dependent on the hydrologic regime of the mountain. So that really highlights the importance of this methodology of bringing together ecological and hydrological monitoring and really integrating those monitoring methods to make sure we can understand those interactions. But critically, without the long-term monitoring at Springbrook via this Critical Zone Observatory, scientific monitoring and reporting for the Gondwana Rainforest World Heritage Area is extremely limited. The monitoring data that we collected is needed to inform local government decision-making, water resource planning, national park management, and World Heritage Area management. We're really trying to build this Critical Zone Observatory by adding on to the monitoring that we've already started at Springbrook, so building on those existing monitoring projects, integrating with other networks, so working closely with Tern and working with the Critical Zone Observatory network, and then really continuing our focus on co-design, so working very closely with our Indigenous and community collaborators to continue to build this monitoring and reporting framework, and making sure that the information is still relevant for community and for all of our stakeholders. So we're going to keep this really strong focus on communication and science to policy translation, and make sure that the data is available for our partners and the key stakeholders involved. So a few key objectives for our project that we're really just starting to develop at the moment. So we're starting to develop the monitoring network. We're focusing on some endangered species and monitoring at different elevations. And the objectives of the project are to incorporate First Nations knowledge in targeting environmental monitoring and reporting, assess temporal and spatial trends in ecohydrology, determine the impacts of cloud moisture and precipitation on soil moisture, groundwater and tree water uptake, assess the spatial and temporal trends in hydrochemistry at Springbrook, combine eDNA and ecoacoustics to investigate the presence and distribution of indicator species, those that are threatened or sensitive to ecosystem change, and finally to develop ecohydrological report cards and 3D visualizations for community education and science to policy translation.
Good afternoon. I'm Siddeswara Guru. I'm the program lead for the Tern Data Services Analytical Platform. So today I will talk about the process we use to make all the material samples collected by Tern available and accessible. So this is a collaborative work between our team and then the Tern surveillance team. So Donna has given a pretty good context about it. So just to highlight, so the surveillance monitoring team, as part of their systematic survey, collect 5 different samples, 2 on the vegetation, and then the 3 on the soil samples. It is available on a big shed in the University of Adelaide. So most of the data is collected from since 2011, and it's available, archived, everything. So the main objective is to basically how to make this available to the wider community so that it can be discoverable, and then the accessible, and then so that it can be requested for use. So that was the main objective. So we have a data integration platform called EcoPlot. So we integrate the systematic survey data, observation data. So as part of this, we have published all the data set from the surveillance monitoring, from the ecosystem processes, as well as the Queensland government, the long-term ecological research network, 3 Savannah Park, and then we are in the process of publishing the biodiversity database from the South Australia as well. So we have a data from close to 25,000 plots across Australia. Anyway, samples is also collected as part of a systematic survey. So we integrate the sample search as part of the EcoPlots as well. So now if you go to the EcoPlots, so then you can search for the samples, and you can search for the observation data as well. And then you can swap between the two while you are searching for any of those. So when you go to the EcoPlots, you will get to the dashboard. So this is a sample dashboard. You can search based on the region. So we index every site for the Ebru region, sub-Ebru region, NRM region, state and territory, local electorate, anything, about 5-6 of them. So you can search based on them, and you can search, currently we have samples only from the surveillance monitoring, so data source will be the surveillance monitoring. And you can search based on the sites, and you can search based on if there is any repeated measures as well, repeated sites. Then you can search based on the samples type as well. So the main thing, the harmonization point is the sample type. So if you search based on the sample type, and then you will get a result of the, what is the samples available. And then once the samples are available, so when the samples are available, you can look at the samples and you can select what samples you want to do. You wanted to know as well. So there is a IGSN there. If you click on that one, so you can see the complete details about the sample. So IGSN is the International Geological Serial Number. It is a persistent identifier for the physical samples. It was started about 10 years back to provide a PID for the geological data, and then later it has extended to any physical sample. So whatever we have published here, it has got a IGSN number. So what it helps us that it uses us to have a persistent identifier for every samples, and then it's easy to share and then access as well. As long as you have the PID, you can access those samples. So you can look at that one and you can select, this is a page for each of the one. So if it has got an image, it will show the images as well. And then it has got all the related contextual information. So we have a standard metadata as well as we had a bit of a contextual information based on the feedback from the surveillance team. So the fair bit of information is available as part of this page. So this basically helps you to not to go to the observation side of the things, to look at the same data set. And then once you decide, look, these are the samples you want, you select that samples and then request for those samples. And then you put some details, so it's for your own good. And then you request for those samples. So as Donna said, you need to basically put up an expression of interest to access that sample. So we ask a set of questions. So once you finish that one, you submit that. So it will create a package for you. Once the package is ready, you get an email. And then you can download that package for your reference, and then that email, while you get an email, it will be emailed to the Adelaide team as well, so that they will start processing your application. So once after that, Adelaide team will start contacting you to understand the process, and then to make sure that you get the samples as well. Yeah. So the salient features of this one is that basically the one place to come and access all the samples available in Tern. So currently we have published around 129,000 samples. So as I said, it's a curated samples. Whatever they said, it's curated, available, ready to make it public. So those are the samples we have published. So one of the things what we have done is that we have minted the IGSN for each of the samples. So I think we are probably the first project to mint a IGSN for the vegetation samples in Australia. Nobody has minted a PID for the vegetation samples. So what it helps us is to standardize basically the samples, how we describe the samples, how the samples can be shared, and then even for the user's perspective, you just need a PID to share with any of your colleagues. You don't have to copy everything. That helps. So if you go to the ecoplots, a lot of samples don't have a digitized image, partly because the team is digitizing a lot of samples still. So as we get those, that will be ingested into this one. So we generally ingest every month. So every 6th day of the month, we get a fresh ingestion from the team. So we ingest, it's a monthly ingestion. So in summary, so we extended the ecoplot to make that available, which is available to make samples available as well. So you can go to the ecoplot and then search for the samples, and you can play with it, and then let us know what you think, and then happy to take any feedback. Thank you.
All right, is everyone still with me? It's been a long session. So I'm going to circle back to drone remote sensing and the role it has to play in ecosystem monitoring. So I'm Alice Robbins. I'm a research associate in remote sensing at the University of Tasmania. So I'm going to be talking about a project that I've been working on for the last year and a half. It's called NatureScan. So this is a collaboration between UTAS, the University of Adelaide, Tern, and it's led by Professor Arco Lucier and Associate Professor Ben Sparrow. So NatureScan is funded through the Department of Climate Change, Energy, the Environment and Water Innovative Biodiversity Monitoring Grants Program in the context of the nature repair market. Broadly, our project seeks to advance the use of drones in advanced biodiversity monitoring, specifically consumer-grade drones, and then we're fusing that with expertise in field ecology, AI, and remote sensing analysis. I'm really going to be focusing today on the collection and processing protocols that I've been working on for the last 18 months, specifically for the DJI Mavic 3 multispectral drone. So you can see the specs for that drone up here. It's a pretty neat little unit. It's about $5,000 if you don't buy it with the RTK, and it's got an RGB camera, a 4-band multispectral camera, and it has this little hat you can install. Oops, let's go back. This one here, and that's your RTK module to enable that centimetre level positioning. So really a lot of this work is focused on developing protocols that can be used for really scientifically sound and robust kind of collection and processing parameters across different ecosystems, so that we know that any kind of analysis-ready data products that we're looking at are returning measures of ecosystem structure, function and composition that are actually true change in that ecosystem and not a change as a result of your collection or processing parameters. We're also really looking to lower the knowledge barrier for this cost-effective drone setup to encourage the uptake of drones for ecosystem and biodiversity monitoring. So I'm just going to cover kind of the first 3 stages of your data collection. So that really starts with like a pre-flight desktop assessment. So our protocol addresses this, and that's just thinking about things like your you're planning your flight mission, so, drawing a KML on a GIS and bringing it into your controller, that'll give you an estimate of how long that will take. It's really helpful for planning for things like how many batteries to take. And then even just thinking about the time of year that you're heading out there, are you trying to capture any relevant phenological changes? Right now in Tassie, the sun angle is really low, so the data quality is actually quite poor. So we kind of address those recommendations as well. It's really important to take into consideration topographic change because that will impact your image overlap. I'll address that a bit later. And so that carries through to your processing and kind of returning really robust data products. And then we kind of just outline a pre-field work gear checklist so you don't end up all the way out there and you've forgotten something super essential. So you're out in the field. We've also written some pretty good parameters about your data collection. That's mainly addressing things like your overlap, so your forward and side overlap. And that, yeah, as I said, I'll get to that with the processing. Thinking about your flying height and your pixel size. So if you're out there and you're trying to capture a flower that's 1 centimetre in diameter and your pixel size is 2 centimetres, go home, there's no point. So, redo your parameters. And then thinking about your vegetation structure and how that impacts, again, your overlap. So taller vegetation will reduce your overlap, so taking that into consideration when you're out there. So you've done all that, and now you get to fly the drone. The biggest thing you have to worry about here is usually eagles. They really don't like the Mavic, so keep your eyes on the sky. And then, happily for me, my colleague Juan has already covered this, but really thinking about your metadata and backing up your data once you're back in the office, so pulling that KML off your controller. That's really good for any kind of repeat monitoring, so you don't have to do that whole pre-flight assessment in the future. Just pull it straight off your metadata that you've backed up safely somewhere. Keeping note of environmental conditions. The sky condition will impact your data quality. And then you can also pull the raw satellite data off the base station, post-process that, and then you have a precise position you can use for any repeat monitoring in the future. So a lot of the work I've done is looking at processing this data. So once you have it, you need to process it. And so we've really focused on how we can do that to optimize the parameters specifically within Agisoft Metashape to return robust representation of different ecosystems, really focusing on 3 things. So the geometric alignment between your RGB and multispectral sensor, the impact of the orthorectification surface on your final model, and then your spatial accuracy. So just a bit of a brief background on what's going on this screen. So structure from motion, basically, you use that overlapping images. So you can see that here. That's where your overlap's really important. So it's going to go through and look for features that match within them, tie them across the scene, and return what we call a tie point cloud or a sparse point cloud. So we then also, in our workflow, we densify that point cloud, so it's just building a dense point cloud. We do that because we don't have LiDAR, so we're really interested in what kind of structural metrics we can pull out of this. And then you do your orthomosaic generation, and you get to your analysis-ready data product. It's quite a lot of information, so I'm just going to break that down into what that actually looks like. So specifically here, I'm testing the geometric alignment between the sensors and also the XYZ precision, and just kind of how your Metashape parameters impact that. So yeah, just lots of testing, lots of doing the same thing over and over again and looking for slight differences. So if I just show that here, this is me processing the 2 sensors separately. So one chunk is just your RGB and one's the TIFFs. If you just keep an eye on this branch here, you'll notice in this false colour composite, there's quite a substantial offset between the RGB and the multispectral sensor. So what we've discovered is that, so keep your eye on that same branch. You can tell Metashape basically that it's a multi-camera setup and to process all the images as a single image stack. When you do that, what we've found is we don't get that offset. So just showing that again here. There we go. So you've got about 20 to 30 centimetres, but this one here is pretty much perfect. We have found that this offset is a lot worse with taller vegetation. So when you get to tall eucalypt forests, you almost get a complete shift in the canopy, and you really don't get it with the multi-camera setup. We are still testing aspects of this, but so far it looks pretty robust. And that's just really important for ongoing ecological monitoring because you want to know that everything's in the same position. And yeah, if you have your multispectral sensor but things aren't lining up, it really kind of plays into your ecological monitoring. So as I've said, you can then take that tie point cloud that you get from your image alignment and build a dense point cloud down here. This kind of comes into play because we're looking at now at your orthomosaic generation, so taking all of those overlapping images and stitching them together. This process corrects for distortions, and you basically get a continuous image of your study area that has been corrected to a constant scale, and you can use it for direct measurements. In this world, there's not really like a standard protocol on how you generate your orthomosaic, and what I mean by that is some people use the tie point cloud, some people use a dense point cloud, some people use LiDAR. You can build a model, which is a polygonal, 2.5D model off those point clouds. You can export your point cloud and classify ground points and bring it back in. You can do anything you want. My main point here is that it does impact your orthomosaics, so you kind of need to know the impact of that, and I spent a fair bit of time last year looking at this. So I'll show you what I mean. So what we've got here is your digital elevation model, so that's your ground points classified in your structure from motion point cloud compared to a digital surface model, so it's still got those elevation features. So you'll notice here that it actually impacts the size of the canopy. And again, this is more profound in higher vegetation. So when we have like short little chenopods, it doesn't really matter. The mallee trees, not so much, but when you get to your tall eucalypts, the size of the canopy will change with your orthorectification surface. It's obviously an issue. If you're going to do repeat monitoring and use different surfaces, you need to know that this will happen. Not only does it affect the size of the canopy, it also makes really ugly orthomosaics. So your digital elevation model is this really beautiful canopy here with minimal distortions. But what you get in the digital surface model is a pixel smear and distortions, where basically in the orthorectification process, it's hit at a really abrupt change. And so you get this kind of smear when it tries to cope with that, which, yeah, it's just super unsightly. I don't really like it, and is also an issue for your ongoing kind of ecological monitoring. So what we're recommending with our workflow, because as I've said, you can classify those ground points, it's really difficult to do in Metashape well, and it's really time consuming to pull that structure from motion point cloud out and do it in something like LIDAR. What we recommend is generating a model using that dense point cloud and then applying a smoothing factor, and that emulates a digital elevation model. Surprise, we found that the model, so basically you can choose, like in Metashape, you can say how many faces you want. They have a parameter, it's low, medium or high. Your smooth value is kind of arbitrary. We do 20, 50, 100, 200, I've tested a lot. We've found that those values matter. So I'm just going to do a little bit of a spot the difference here. So this is a low model with a smooth value of 20. This is a medium model with a smooth value of 100. Is it going to work for me? Yay. So if I go back and forth, just see if you can notice if anything's changing. And so what you notice with this is in the low model, you end up getting like a duplication of features. It's also called ghosting. So these logs up here have also been duplicated. So I'll just go back and show that again. There we go. So we have found that with a medium model with a different smoothing value dependent on the, again, height of the major vegetation, you don't get this kind of ghosting. This is, again, really important because if you're doing like a coarse woody debris count for your ongoing ecological monitoring and you're getting, misleading counts of how many logs there are, it's a problem, so you need to know. So you get to this phase with your analysis-ready data product. So this is just the Mavix data. So we are looking at using the RGB orthomosaic with a co-aligned 4-band multispectral orthomosaic, and that structure from motion point cloud to really look at how we can measure ecosystem structure, function, and composition, and then how that informs, how that informs remotely sensed biodiversity products. I better hurry up. So just to show what that might look like, you can look at things like canopy cover, fractional photosynthetic cover, NDVI, and then you can use that point cloud to look at things like, again, canopy cover, structural variability, and your canopy height. So leave that there for 1 second. And then just to briefly touch on some other work that we've done in the project, we have established a series of restoration regeneration sites. We've been working with Bush Heritage Australia, and last year we mapped one of their new reserves, Evelyn Downs, where they've recently destocked and took 5,000 cattle off that property. So when I was out there last year, we mapped quite widely across these ecological communities, but we also mapped a series of dams because they are planning on using that structure from motion point cloud to basically do some hydrological modelling. Brilliant. And then just in summary, obviously drones are a super powerful tool for ongoing ecosystem monitoring, but you do need to understand the impact of your collection and processing protocols. We've done that work and intensively tested this for specifically the Mavic 3, but it does kind of carry over to other platforms. We will be making these protocols publicly available. They're currently in their final review, but if you are interested, please contact me. And all data we collect for this project will be made available along with the protocols on the Tern data portal eventually. And if you're interested in keeping up with the project, there's the QR code for our website there. Thank you so much.
don't want to stand. I'll get up here. I don't want to stand between you all and afternoon tea, but we do have some time for some questions. And if the speakers could stand up here. Unfortunately, we don't have enough chairs, even though I suggested bringing in some shooting sticks, those three-legged things.
Do you want a live thing?
I've got it.
Okay.
Oh, thank you.
The other thing is we have a hotel key.
We have a hotel what?
Hotel key, so if anyone's lost one, if they could come.
Oh, yes, good. Okay. While everyone's getting up, we have a message from Jill, which is that someone has left a hotel key somewhere. Just look and make sure that you've got the key to your room. If you haven't, there's Jill. So please go and see her, possibly in morning tea. So thank you all for standing. What a wonderful session. A brief summary from my point of view. We saw some great examples of scaling, bioacoustic data, groundwater at small and large scales, the plot monitoring that's been done both locally and across nations, a great variety of tools and versions of those tools. The last session, the last talk by Annie showed us very much how to, here's a tool and we can do it in, so we can manipulate in several different ways, and so did Dave do that. And of course their refinement to make things even better and work even better for the purposes of the user and the purposes of the monitoring. Of course, one of the things that I'm fairly stunned with at the moment is the enormous data availability, shouting out for the sponsor, that is now available in Tern. Not just the availability, the bulk of it, but also the variety of data that's now coming through Tern, both in the big shed in Adelaide, but also in many other ways, as you've heard, and particularly making those data more available through labelling, through all sorts of things. And of course, integration across sources. So integration that no one really operates alone. No one source actually operates alone. All to determine the greater answer, namely ecosystem integrity, its ecosystem status and effects of change. So after that, so I was particularly impressed and a bit dazzled because it got a bit, and then it got better, and then you'd think, oh, I can't deal with another one, but then it was brilliant. So they were all good and kept me awake for sure. So I'm sure it kept you all awake. So we've got a few questions. We might ask Brady, the newcomer to the country, here, putting you on the spot. What do you think about the potential to use ground-penetrating radar to inform us of subsurface geomorphic changes impacting anthropogenic surface processes, so our stuff, and accelerating their impacts? And Kalisha asked that question, Brady. Sure. Thank you.
Is that on? Yeah. Yeah, it's a great question. I have been using ground penetrating radar. It's a smaller scale tool, so it's probably really good for soils. So I have a student at the site I showed you. I didn't get a chance to go to the other side of the watershed, but the reflections actually match up where the knocking pole survey is. So if you have anthropogenic change or you get changes in erosion rates, you can probably map out that soil. So my answer is it was a pretty broad question. Probably, right? We probably sit down and do it. The structure seems probably pretty relatable to that.
So we can tell when we drill holes in things that we can actually change a lot more than we think. Yes. I think, oh yes, you might have to pass that round. David McJannet has a question. And thank you to the speakers up there. It's nice to have you all there. It's great, so yeah. Okay, so for David, we have a question again from Kalisha, who's been very good. Do you think we could potentially use the Cosmos data to calibrate mixed pixel effects within water observation satellite imagery? So, yeah, to more accurately define water bodies. You know how you can use... We heard about it from someone, from the different sorts of satellite imagery.
Okay, just
bands you can use.
Yep.
So could you complement cross-source, I think is the idea.
It'd be fairly tricky.
To more accurately define water bodies.
Was it more accurate? No, probably not, would be my answer to that. It's you get a mixed signal of, because it's a circular footprint, you couldn't use a Cosmos sensor to be able to determine the edge just because of the way it works. It's a central measurement. It doesn't pick up edges. It would be very tricky to do. I think there's probably better ways to do that, probably with satellite imagery, some of the higher resolution stuff.
Yeah, so Kalisha. Sorry about that. It's a good idea, but maybe we could pursue that, the other option, some other way. For Cho-Ying, here you are. Thank you for your talk. Has anyone used the bird vocal activity signal to predict implications for pollination in the tea plantations or surrounding biodiversity? And Professor Morris has asked this question.
I think the reference before is about China tea forest mosaic. But in Taiwan, it's something different from China's tea plantation. So I think it's great to use the soundscape indicator to estimate the biocomplexity or diversity in Taiwan's tea plantation. Yes.
Okay, so you could assess how monoculture the plantation is versus can you because the birds that you're listening to will help predate any pests too. So I suppose that's important. Yeah.
Yes.
Thank you. We don't have terribly many questions. I should probably have got one for mangroves or whatever. For David again, and this is from Beryl, is such information about the measurement footprint provided as part of the data set as there would be useful information for scale matching with satellite data? Same question, the matching up with satellite, and how does that, can it work?
Definitely. So we don't publish the footprint, the spatial footprint, but we do publish the depth footprint to give that information. So we could easily publish the spatial extent, but it doesn't vary that quickly, and it's at such a scale that it's past that small scale variability. So we're at that matching to satellite scale anyway. So it doesn't give us that much extra information, but it's possible to do it. Yes.
And Elon's satellite isn't close enough, I take it. Yeah.
Yes, it doesn't help.
It's not like spotty miles or something. Okay. Finally, what sort of, I suppose this could be for everyone, for many of the tool providers, but what sort of outreach or educational access workshops have been delivered or are to be delivered in regards to the it's happening again, Cosmos network in particular, but for everyone, to increase data use? So maybe we'll avoid David answering that one. And would anyone else like to talk about your outreach activities? And of course, I am interested in Lucy. Are you here, Lucy? Yes. Yes, because you've been working on the a very populated hill. No, human population. Yes. So outreach.
Yeah. So far we've had a few community meetings where we've been sharing the type of monitoring we're doing, and in our next phase we're hoping to start sharing access to our data through our visualization platform. So it's definitely our next stage, but it's a really important part of our current project, and it's something that we want to keep building on as we go forward and trying to make sure that there's enough interpretation and explanation of the data sets as we go. Another thing we're starting to think about is maybe trying to have an education centre up at Springbrook where we can share the data and the information we're collecting, maybe some of the recordings we're collecting as well. So we've got plans to grow that,
Why am I not surprised? But I think what the question was about the data. Yes. Oh, we've got to stop. No, I'm looking at it. I have 2 more minutes. Was the data, sharing data. So we've talked, Guru has shared data. That's Tern's idea. Cosmos, have you shared data? What about Caitlin sharing data? Anyone who's involved with Tern, I suppose we're obliged to share data, aren't we? But yes, so the outreach workshops are a really big deal. But yes, for the Cosmos, data pickup by encouraging people to use it. Anyway, so we have 1 minute.
We haven't run any workshops, but the data's there and it's fairly straightforward hourly soil moisture that anyone can access in easy form. So.
There you are. I will. We won't say, trust me, I'm from Syro. No, anyway. Thank you very much, everyone, for listening, and thank you to the speakers who've been fantastic, all of you, and thank you all together. Okay, afternoon tea. All right.